Good evening, class 10, final practice on heredity practice questions. Question number 1. Why did Gregor Mendel choose peas for his experiment and not others? Why? You have to write, it is easy to find varieties of peas everywhere in all seasons. It is very easy to study the life cycle of peas, about 2-3 months. Different varieties of peas are found, like tall and dwarf peas, red and white flower, yellow and green seeds, yellow and green pods, terminal pod and axial pods, wrinkled and round, etc. Different varieties of, depending on different colors, shape, size, colors of flowers, colors of pods, etc. State Mendel's laws in 3 points. First law of dominance, means a dominant character suppresses a weak character. Law of dominance. Another law of purity of gametes. Second law of purity of gametes or law of segregation. It means gametes are pure and they don't mix each other. He concluded by doing different experiments, Mendel concluded the 3 points. Number 1, law of dominance. Number 2, law of segregation or law of purity of gametes, and another law of independent assortment. Two contrasting characters don't affect each other, they are independent. Question number 3, distinguish between dominant and recessive. Just by the word dominant, it is a strong character which dominates another, and it can be seen. Recessive character, it is hidden, no देखिने, but there is. It can be expressed, it cannot be expressed. It is hidden. But recessive character can be seen in the F2 generation. Monohybrid and dihybrid cross. Monohybrid, it is the cross between only one type of contrasting character. Only height, like tall and dwarf, or color, or shape of the seed, etc. Only one contrasting characters. Dihybrid cross, it is the cross made between two contrasting characters. Monohybrid cross, final ratio is phenotype ratio 3 is to 1, genotype ratio 1 is to 2 is to 1. Dihybrid cross, see, in this case, law of dominant, but here law of dominance is not proper. Final ratio 9 is to 3 is to 3 is to 1. Phenotype and genotype, it is the external character which can be expressed outwardly. Genotype, it is the internal structure of the gene, it cannot be expressed outside. Phenotype ratio in the second generation 3 is to 1. Genotype ratio 1 is to 2 is to 1. Variation and mutation. Variation is continuous change. Any change in two or more organisms. Variation is continuous, generally environmental variation. According to Charles Darwin, variation is the cause of origin of new species. Mutation is a sudden, unexpected, drastic change in the genetic structure. It appears all of a sudden and it is operational in all generations, next generations. For example, 6 fingers. According to Hugo de Vries, mutation is the actual cause of the origin of new species. Question number 4. Define hybrid variation. Hybrid. A hybrid is an organism formed from two genetically different organisms belonging to the same class, same phylum or group, and same species. Variation, heredity, transfer of parental characters, gene, thread-like structure found in the chromosome. It is responsible to transfer heredity character. Genetics, systematic study of variation and heredity. Genetics, dominant and recessive characters already. Question number 5. Draw the monohybrid cross chart up to the F2 filial generations when a black capital BB and tall TT, two characters, color and height. Black and tall dog is allowed to cross with a brown small b and dark beach TT. See how to draw. See the solution of this is let B for black, small b for brown, capital T, tall, small t, dwarf. Dwarf means short. F1 generation. Here, it is the not the monohybrid cross. It is the dihybrid cross. Sorry, it is dihybrid. Monohybrid cross single, but here dihybrid cross. Let B for black, B for brown, capital T tall, small t dwarf. F1 generation. F1 filial generation. How to write down F1 filial generation? BB like this. You have to write this way. Capital small. Not capital B, capital B. Here BT, black and tall, or tall and black. You can write anyway. Tall and black are BT, capital small and capital small and small. Black and tall, black and dwarf. Tall and brown, brown and dwarf. See, it is better to draw only the F1 filial generation. F2 filial generation is not necessary in this case because it is not in the course. Only F1 or monohybrid crosses. Cross, you have to read monohybrid cross only, not dihybrid cross. Question number 5. Question number 6. Now another. Question number 6. It is draw the monohybrid cross chart up to the F2 filial generations when, say it's better to write green, when a green pod, a pod, green pod GG P plant is cross pollinated with the yellow pod small g plant. हरियो कोसा हुने केराउ र पहिलो कोसा फल्ने केराउलाई क्रस पोलिनेसन गराउँदा what is the result? What result can be found? Solution. But in this case, if the green is dominant and yellow pod is recessive. But in case of seed, yellow seed is the yellow seed capital Y and green seed small y. See. But in this case, pod, not seed. So it is better to write down pod. Let capital G for green pod, small g yellow pod. हरियो कोसा पहिलो कोसा हरियो कोसा फल्ने केराउ पहिलो कोसा green podded seed plant and yellow podded seed plant. Here green is dominant in this case and yellow pod is recessive character, weak character, which is hidden. In the first F1 filial generation, what result can be obtained? Capital G and small g. Here, gametes are pure and they don't mix each other. See, you have to know it. Capital G, small g. Green pod and yellow pod. See, cross, capital small. Again, cross. Okay, here, this and this one. This. And what is what result? Capital G, small g. Again, capital G, small g. First, cross. Again, capital G, small g, capital G, small g. What is the result? 100% hybrid green pod. 100% it is called hybrid green pods. Not seeds. Seed only, pod only. Green pod. In the F1 filial generation. What generation is this? Kun generation? Capital small. If there is capital small, it is first F1 filial generation. F2 filial generation. Now cross. Capital G and G. See. Now hybrid green. Now cross. See. Capital and capital small capital small. What is the result? Capital G, small g, capital capital and small, finally small. What is this? One pure green pod, two hybrid green, and one is pure yellow. See, one pure what? Pure green, two are hybrid green, and one is pure yellow seed. What is the final result? Final result is done in this way. First of all, capital G, small g, 100% hybrid green pods are found. In the second filial generations, capital G, small g, capital small. What is the phenotype ratio? See, let me write down phenotype ratio here. Already written. Phenotype ratio is 3:1 because 3 are green, pure green or hybrid green pods. 3, 1 pure yellow. शुद्ध पहेँलो हुने खालको. genotype ratio is 1:2:1. Why genotype? Why 1:2:1? One is pure green, not hybrid. One is pure green, two are hybrid green. Externally we see hybrid, but it's mixed. Hybrid means mixed type. Pure green 1, pure yellow 1. Hybrid green or mixed type of green 2. So 1:2:1 genotype ratio. Okay. Next time I will do other exercises, other lessons, other units. This was for heredity and variation. Practice more. Go through the book. किताब राम्रोसँग अध्ययन गर्नु होला.